Thank you very much. My name is Steve Laramie. I'm a medical oncologist within FDA, and I'm really honored to be here in front of so many people who have worked so hard to improve the lives of patients with GI cancers. So I was asked to deal today, or present today, on how to deal with the explosion of new treatments. So I think this topic can be tackled in so many ways, so I'll give an effort to do the first job at it. How do we move forward? With this? Oh, okay. Oh, okay. Thank you. So I thought I'd start out for starting in 2011, for about a 6-year period. I was I was exclusively working with GI cancer drugs, and so I thought I'd list all the drugs that FDA approved, not including some of the vaccines, since 2011. So clearly, there's many drugs approved here. Many of them have been transformative for some of the patients with different cancers. However, clearly the approval of new transformative drugs in GI cancers has appeared to lag behind some of the other tumor types. So for GI cancers, some of the drugs we approved include 3 drugs that affect the VEGF pathway and produce modest improvements in survival in patients with colon or gastric cancer. We approved TAS-102, which also is a modest improvement in third-line colorectal cancer. And we approved lutetium-177 dotatate for neuroendocrine tumors, which has been used for years, decades in the Netherlands, and probably has a bigger effect for patients with neuroendocrine tumors. However, we're still trying to learn how to sequence it. Finally, I highlighted nivolumab and pembrolizumab, which have received accelerated approval for gastric cancer and hepatocellular cancer. But probably the biggest effect of these 2 drugs is in patients with MSI-high cancers, which we, for the pembrolizumab, received the tissue-agnostic approval. So why has it been tougher in GI oncology? Well, it's, biology is most likely their main reason. So few of our patients with GI cancers have druggable targets. Many of the patients with GI cancers have a lower mutation burden compared to patients with melanoma or lung cancer. So immunotherapies may be harder to use for these patients. Is investment with companies different? I don't know. I don't have a sense of that. Of course, biology is paramount. We have to keep continuing on building what we've learned about the biology of these tumors, what Dr. Tabanero has done and Dr. Tejpar in Europe have done for many years. So despite what I've said in the previous slides, clearly progress has been made in different GI cancers. This lists some of the items that we've, as a community, have made progress on in the years in pancreatic and colorectal cancer. Just in this meeting, we've heard a positive trial in HCC, as well as Dr. Kanwa's positive trial in adjuvant, which is adjuvant pancreatic cancer, which will really make a big difference for patients. So in the red boxes, I just listed some of the applications and items which I've had the opportunity to work on while at the FDA. So regulatory progress. I thought I'd transition a little bit to how we've gone to this point as an FDA with some of these new drugs. When I started at the FDA, my review work really focused on cytokines and monoclonal antibodies regardless of indication. My first molecular entity that I worked on was ofatumumab for CLL. It was a very different agency back then, and what Dr. Paz has really done, I think that the most important, there's so many things that have happened at the FDA in the last 10 years, breakthrough, et cetera, but what he's really done is made specific teams working on disease expert teams in the different fields like GI cancers, breast cancer, lung cancer. And I think that's really made more of an academic atmosphere at the FDA. We're writing a lot more, we're giving more talks, we're being interactive with the community, with ASCO, ACR, hopefully with ESMO. And I think that really has made a big progress in how we deal with biology, how we deal with drugs. And coincident with that approach, we've seen a profound understanding of improvements in biology for different tumor types, and some drugs which are transformative and really have helped some patients. So from a historical perspective, in the 1970s, FDA approved drugs mainly using tumor shrinkage as an endpoint. So at that time, there were limited therapies available, and at the time, we granted regular approval for some of these therapies, mainly cytotoxic drugs. In the 1980s, a change in this interpretation occurred. So we saw limited response rates for some of these cytotoxic drugs, and they were toxic, and often they did not translate into benefit. So it was really a shift at that time to overall survival as the primary endpoint for approval. So and then this started to happen, and This was a paper in 2001 for imatinib in CML, which really transformed the landscape of treatment for this disease. Profound change in knowing both the biology and a treatment for that biology and that disease. So it took many years for that revolution to really occur in solid tumors. So that was 2001. Now we're seeing this in some solid tumor types. So we understand the biology, we have enriched populations, strong basic science, and durability of some of these responses. So these are 3 different applications that I've had the pleasure to work on. One for brigatinib in lung cancer, for ALK-positive lung cancer, one for BRAF-positive anaplastic thyroid cancer, and one for MSI-high cancers, all with large response rates and durable responses as well. So looking closer at how we look at response rate as an agency, there are multiple variables for response rate. We look at tumor burden location. Again, a small pelvic lymph node that responds is different than a large local response in a patient with anaplastic thyroid cancer. Look at available therapy, unmet need. Are there complete responses? The durability of these responses. How many patients have the tumor reduced but less than 30%, as well as disease prevalence would be considered. Here's an example of where our tumors responding. So we've given full approval for certain dermatologic cancers who have had profound responses in disfiguring dermatologic lesions. The example on the left is vismodegib for basal cell cancer. The example on the right is denosumab, which I had a little bit of chance to work on. These are rarely local destructive tumors. So the patient right there has a sacral lesion and perhaps would have had to have an extremely morbid surgery to remove that tumor. So ultimately, the ultimate endpoint design, end trial design that should be considered for trials, it really depends on context. So equipoise is important for the ethical context of randomized trials. So the example on the right is crizotinib in lung cancer, which had a close to 70% response rate, median duration of response of 18 months. It's a rare population. There's no other therapies. So we granted that regular approval. Conversely, the situation on the left that you know all well, it has a regorafenib, it had a 1% response rate, but ultimately it did demonstrate benefit with an improved survival effect in these patients. However, there was equipoise to conduct that trial where there may not have been for the ROS1 positive lung cancer application. So randomization still has value. So again, everything is context dependent. So the example on the left is where we used overall survival as an endpoint. You all know this trial very well, TAS 102. The study did demonstrate a PFS effect, but it was a modest PFS effect, and the fact that it was able to demonstrate a statistically significant effect on overall survival made the application much stronger. Whereas on the right is lutetium 177 dotatate. This is a slower growing tumor. You had a very large effect on PFS, and requirement to demonstrate a survival effect would have resulted in delay of approval for many, many years. So these are some of the guiding principles moving forward. The best access for patients, of course, is approval of an effective drug and affordability, and I think we'll hear a lot about that in the next presentations. I'll mostly touch base on what's in the red box for investigational agents, focusing on more inclusive trials, better trials, and learning from the past. So more inclusive trials. One of the hot topics I'll highlight moving forward is that we need to think carefully about who we enroll in trials and where appropriate, increase enrollment into clinical trials to more closely mimic the patients who will receive the drug post-marketing. So these are some of the areas we focused on as an agency to improve eligibility: CNS, performance status, comorbidities, HIV, autoimmune diseases. The example on the right is an application for alectinib where these patients with ALK-positive lung cancer frequently have brain mets, and the company enrolling these patients greatly sped up development of the drug because they did enroll these patients, as well as it showed that the drug worked in brain mets, allowing for labeling claims in this population. So here's a FDA analysis of colorectal cancer that focused on the comparison of age of patients enrolled in colorectal cancer trials in applications submitted to FDA compared to the expected population of U.S. incidence of colorectal cancer. And clearly patients enrolled in clinical trials are younger than, and I think we all know this. And this is an older slide, but it's probably still close to true, although I don't know if the data will completely hold up, but this basically shows that if the prediction is if eligibility criteria are relaxed due to comorbidities, that we'll probably be able to enroll a more representative patient population, at least based on age. So now I'll shift to trial designs. And I think it's important to design a trial thoughtfully to ensure we enroll the appropriate number of patients in each trial. So clearly, novel designs are facilitated by biomarkers. So there are, of course, there are trials that will test multiple drugs at the same time. In one cancer, we have, the MRC Focus 4 trial in colon cancer. There's a planned Precision Promise trial to be intended to be run by PanCan that they publicly announced. There's also trials that can test multiple cancers in the same trial, like NCI Match or expansion cohort trials. There are registries or real-world data, as well as there's seamless designs, which I'll show you in a second, which are, in essence, a drug development in a single trial. So here is what we call a seamless design. So this, you can see it's a very complex study. This was pembrolizumab for the initial approvals for melanoma and lung cancer. It was a study that enrolled over 1,000 patients. You can see there are multiple cohorts in this trial, both with single-arm cohorts as well as randomized cohorts. Very complex. Although these might be appropriate in a limited number of circumstances, there are also real concerns regarding oversight of such trials. How is safety being followed throughout the trial? If not done thoughtfully, it really has the potential to delay development and increase cost if you're just enrolling patients without a lot of thought. So although Keynote-1 was a complex trial, it didn't start out that way. So I think the IND was submitted in 2011, and as you can see at the beginning, there were relatively few patients that were enrolled. Started getting an amendment right before 2012. I think at this time, we're starting to see that there's real activity of this drug. And then as you can see, it got really complex with multiple different amendments, different arms, and a rapid expansion of enrollment. I think what's important here is that we did have lots of interactions with the company in this case to facilitate the development of the drug. It was a breakthrough therapy. It was an important drug with an unmet need for different patients. So this was appropriate in this case, but it's not necessarily should be the de facto start for most drugs. So here's our perspective on seamless designs. So these are implications of a full drug development program within a single protocol. There needs to be the appropriate level of IRB scrutiny of these trials. Where appropriate, we need to have meetings between FDA and EMA and these companies. And it may be more difficult in these trials because, again, you don't have the clear demarcation of end of phase 1 meeting, end of phase 2. It's just a single trial. You want to make sure the size and quality of the safety database is adequate and that the study could be adequate to support global regulatory approvals, not just in one jurisdiction. And with informed consent, the consent probably will need to change from when you start the trial compared to when 600 patients are enrolled. Oh, I'll go back. I think what's important is that there needs to be real justification for the sample size in these trials. So I think all too often now we're seeing 300 to 600 patient trials submitted first in human trial, multiple different cohorts and combinations, and there's not really a lot of thought into the design and what questions are actually going to be asked to obtain relevant data. And if that's not the case, you're really just, we're not doing the right thing, both from drug development perspective and for the patients. Finally, I'm going to transition to learning from prior experience. So this is my tree of publicly reported trials in pancreatic cancer. Most of these were described in a paper by Jordan Berlin and the Pancreatic Cancer Action Network. So I'm going to click on. So all the drugs in the green boxes are drugs that, from what I could best tell, had single-arm data and they moved into phase 3. Most of these had very limited response rates or a limited response rate in combination with gemcitabine, and you couldn't isolate the effect of the drug in combination with gemcitabine. So it was minimal data that moved into phase 3. So rigosertinib was the same. However, it was stopped in early futility analysis, which was probably appropriate. There were 3 drugs that did have randomized phase 2 trials. The top one, CRS-207 GVAX, actually, there's a breakthrough designation granted for that, and unfortunately for that one, it didn't pan out in the subsequent trial. Another one of these 2 drugs was moved into phase 3 based on a positive p-value in a subgroup analysis. So some of the lessons to learn here are don't overinterpret trial results when planning to go into phase 3. This especially holds true for single-arm combination studies, and I've seen it over and over again. We have a response rate, but it's hard to interpret the effect in compared to a single-center study where, seeing a patient at Sloan Kettering is different from, the patients that will be enrolled in a phase 3 trial. So same thing goes for single-arm PFS and survival. It's hard to interpret the data in these trials. And thinking about p-values. So again, a p-value of less than 0.05 is not statistically significant necessarily unless it's in the right context. So the picture, just to give a very loose example of this, the picture on the right is Hugo Lloris, the goalkeeper of France. More importantly for my son, he's a big Tottenham Hotspur fan. He's the goalie of the goalkeeper of that team. If I were to shoot a penalty kick on him, I'd have less than a 5% chance of scoring that goal. However, if most of my friends that are pre-specified try to take a shot at the same time, probably one of those balls would get in. It's not the perfect analogy, but it does apply mathematically that the more times you test, and we see this over and over presented in academic meetings, you'll see a p-value that's less than 0.05. Well, that just by chance, it's likely to occur if you test enough. So I think it's important lessons to learn is move on to the next drug earlier if a drug is not effective. You can expand if there's unprecedented drug effects. And really about enrollment, you have the power regarding enrollment. It's hard for us to stop a trial just for an efficacy reason. It's for regulators. It's really up to the academic community to say, to really think hard about whether you should participate in this trial if it doesn't appear that promising. So finally, I think this was a little bit what I was asked to talk about, the explosion of new cancer treatments, this being the future. At one point, there was one PD-1. I called it Sponsor A. Then they developed their CTLA-4. Actually, the CTLA-4s actually came first, but IDO inhibitor CD40s. Then you have Sponsor B with their PD-L1 or PD-1. Different companies CTLA-4. Third company is chemo. Fourth company is cytokine. And obviously, the combinations are endless. We have probably over 20 PD-1, PD-L1s, multiple different other immunotherapies that each company is developing in combination. And so we have a lot of drugs out there. It's hard for regulators to really have power to do anything about this. Dr. Pazder has suggested to companies maybe they can combine their PD-1s in a single trial, for example, like gastric cancer, but so far industry hasn't been receptive to that idea. But again, with so many drugs, it's even more important to really think carefully about what drugs are actually being developed. And I think as a society, some of these issues that we talk about with endpoints, all these drugs, really what's underneath it is the cost of these drugs. And I think with the number of drugs, I think people would consider it less of a problem if this competition actually reduced prices, but we haven't seen that yet, for example, the VEGF inhibitors and the PD-1 inhibitors. So in summary, in an era of unprecedented drug effect in targeted populations, we need to use the appropriate context-specific regulatory approaches, encourage inclusive enrollment, consider new trial designs, but thoughtfully so. Have a thoughtful use of biomarkers. We need to learn from prior negative trials, and you as the investigators really can control what drugs are studied. Thank you.